Uh, Very good evening. Is it clear? Hello. Good evening. Okay.
Good evening, sir.
So happy Ugadi.
Thank you, sir. Wish you the same.
Okay. 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 Happy Ugadi. To wish to happy Ugadi. Uh, Today we discuss the ecosystem. The remaining type studies are there. Okay. So ecosystem. So these ecosystem is also spin of the Pteridophyta member. This is the division is the Spinopsida. Class is Calamopsida. Order Equisitales. Family Equisitaceae. Genus is the Equisita. Okay. Is it audible? Clear? So
Sir,
the external morphology means this equisetum is referred as a living fossil. Okay. And this one only equisetum only the Spinopsida is living generally remaining all are the extinct. So this one commonly called as a arch tail. So Equisetum is represents only the living genus of the order Equisitae. So this genus only present living genus. Remaining all the species and genus are extinct. So ecosystem is around 25 species are there in the around the world like Australia, New Zealand, and India is also. They inhabitant in cool temperature region only they can grow, and some roadsides in dry places also they can grow. And some Indian species are there: Equisetum maximum, Equisetum elongatum, Equisetum diffusum, and Equisetum dibubum. So these species are grows in India. So this Equisetum is a sporophyte, so which is the herbaceous perennials plant. They have horizontally creeping and subterranean rhizome rooting at nodes and producing aerial shoots which may be branched or unbranched. So this one is the creeping sporophyte plant. Okay, creeping sporophyte plant. And the aerial shoot may be dry up during the summer, but the rhizomes can be renewed through the when the condition is favorable condition. Okay. The aerial shoot as well as rhizome are similar in organization. The plant body is differentiated into nodes and internodes. The whole of the scale leaves from sheath uh, around the stem at each node. However, the number of leaves at each node varies in different species. The lateral branches, if you will be alternating with leaves at nodes. Intercalary meristematic tissue is present to just above the node. So these intercalary meristematic tissue to give the continuous divide to growth of the shoot. So above the node, the intercalary meristematic tissue is present. And the aerial shoot uh, are green and carry on the photosynthesis. The leaves are reduced to scales in the primitive form. The green aerial stems are branched and stabile are borne on the main axis as well as branches. In equisetum pretens The stabile occur only on the main axis, even though are branched. So these further is reduction leads to unbranched green stem terminating cones. So here these aerial shoot means the terminating area to form as a stabile or cones. So here two types of the aerial branches grows on the equisetum, which is the green branched sterile shoot and brown unbranched fertile shoot. So green branched that is the sterile shoot they get involved in the photosynthesis. Another brown unbranched fertile shoots which are bear the stabile. On the terminal region of these brown and fertile shoots, they bear the stabile. These stabales are the reproductive organ in the equisetum. The brown fertile shoots became green and branched after liberation of spores. So after that, again these brown again they became as a green after the spores dispersal. So species which are two types of shoots are below in advance of them. Those are which are in subdistinct branches are there. That is the stem has allowed due to the presence of profound teeth and they very rough due to the solidification. The stems look as if joined at the nodes, mostly due to the conspicuous leaf sheath. The term easily break to the terms to the presence of meristem which are active only for the short duration. So coming to the internal structure of the equisetum, when we take the rhizome, here nothing but root or rhizome. The roots or equisetum is advantageous root in the origin. They are born on the only nodal region of the rhizome. Okay. So here when we take the transverse section of the root, here outer epidermis followed by the cortex and stele is present. Okay. The epidermis outer layer is the epidermis and cortex centrally stele. So the outer layer is epidermis is a single layer. They are produced root hairs. Sometimes they are produced. The cortex is only below the epidermis. Cortex is present. The cortex is parenchymatous near root tips, where the mature portion and the outer layer of the cortex develop into lignified tissues. So here below the cortex is the pericycle is there, significantly absent in the equisetum. Here the endodermis clearly present between the cortex and stele. Okay, pericycle means in normal root cortex, the lowermost layer of the cortex we call as a pericycle. In endodermis, this is the differentiation between the cortex and stele. The endodermis two layer, but only in the outer layer possess the prominent Casparian. So here two layers of the endodermis is present, but the outer layer only they have the Casparian. So the second layer can be interpreted in the pericycle as give as rise to the lateral roots, and the stele is present. The center, which is metaxylal tracheids, is surrounded by the three or four protoxylem points are there. Each protoxylem point is present by a single trachea between the protoxylem points. The area is completely filled up the xylem. So the phloem is there. So here and there are the tetrad or triarch or tetrarch xylem is present depending on the protoxylem point in the stem. Next, coming to the stem, stem transfer section of the ecosystem. The stem mainly shows distinct here epidermis is there, cortex and stele. When we take the transfer section of the stem also, here three distinct regions are there: epidermis, cortex, and stele. So the stem is rigid, hence the section shows ridges and furrows or grooves are there, ridges and furrows. The epidermis is a single layer covered by the silicified thickness are there. Silicified thickness are there. Due to the presence of these silica thickening, the stem is the rough surface. When we feel that stem, it is very rough in condition is there. The stomata are present and the epidermis between the ridges, that is in furrows. Each stomata is surrounded by the two guard cells are there. Okay. The cortex is well differentiated and provide mechanical support to the stem. Beneath the ridges, the cortex may show sclerenchymatous tissues. The vertical rod-like strands of the sclerenchyma are present inner to ridges and they provide maximum mechanical support. The peripheral part of the cortex is in green and which is in photosynthesis activity appear as a bands below the growth. This tissue consists of intercellular space, especially opposite to the stomata. In the inner to the sclerenchyma, there is a colorless tissue called parenchyma. Is there? This portion contains large air-filled cavities called vascular canal, which are opposite to the pharos. They are alternate with the vascular bundle. So, and this is below the cortex is endodermis is cover a steel and followed by the single layer of the pericycle. Central portion is filled with the pith. So the pith is there. So that's why siphonosteel condition is present. So which is by this vascular surrounded by the vascular stand ring is there. These stands are opposite to the ridges and number of stands at an internode is always equal to the number of leaves present at the nodal above. So each vascular bundle has a large water-filled cavity, that is the carinal canal is present. Cortex filled with the air chamber, that one is the valvular canals. Okay. Then this one is the vascular bundle is filled with the carinal canal, water. This carinal canal is conducting channel. A few annual or spiral brackets are present on either side with the carinal canal. The carinal canal is formed from the protoxylem. The outside of the carinal canal, there is a phloem which is consists of the phloem and parenchyma cells are there on either side of the phloem. So here. The phloem and metaxylem is present, which is consist of scalariform brackets. The central part of the pith is performed by a large cavity. The vascular bundles can be considered as endark and collateral condition. The ecosystem stem possesses both xerophytic and hydrophytic features are there. That is the xerophytic condition. Here, silicification of epidermis is present. Silicified silica is deposited on the epidermis, so that's the silicification condition we call that one. And presence of sunken stomata is also there. Presence of sunken stomata and hydrophytic characters here. The presence of air cavities are there. That one is the valvular canals are present in the cortex. and the presence of reduced xylem. These conditions here, there are the two characters are there. That is the hydrophytic characters of xylem. Now, the life history of the ecosystem. To these ecosystem, there are the 20 species are there. So, the each sporangiophore has a stalk and its end in the flattened into polygonal disc. Each is about the uh, 5 to 10 sac-like sporangia arises along the margin of the disc and hanging around the stalk. Okay. The epiphyta maxima, the cones are large and sporangiophores are branched. The development of sporangium is not strictly eusporangiate since the sporangial initial cut of an outer cell which produce a jacket and tapetum and part of the sporangial tissues and inner cell that contributes to only the sporogenous tissue. The tapetum is plasmodial type. The mature sporangium has two-celled thick wall and several spores are there. So, in the ecosystem is the homosporous condition. Okay? Ecosystem is the homosporous condition, and the outer wall is exosporium consists of four spirally twisted elaters also present. The elater shows some hygroscopic nature and which is helps in the spore dispersal. These elaters are formed from the wall but not from sterile sporogenous tissue like bryophytes. In bryophytes, the sporogenous tissue to produce the elaters, but here the wall cell only to produce the elater. So the spores contain chlorophyll according to Campbell. And the spore develop into male and remaining off into female gametophytes. So this spore germinating in the crowd manner produces small dioecious gametophyte where they germinate at the distance produce large monoecious gametophyte. So after these ones to develop as a gametophytes. the gametophyte are prothalli are dorsiventral and are fixed by the rhizode arising on the ventral side. So the dorsal lobes are green and sex organs occur in between the lobes. The antheridia look like those lycopodium and they are embedded in the prothallial tissue. The antheridium developed by the usual method and it produced around 56 ciliated means multiciliated spermatozoids or anthrozoids. Okay. The archegonia have the short neck with two or neck canal cells are there. Okay. So when they are in two numbers, they may be boot shaped or lateral or one above another. The venter is sunk into the prothallus and it contains the egg and ventral canal cells. Okay. The sperm fertilizes the egg form. First cell is the sporophyte, so that is called as zygote. So after zygote, then to develop number of division to produce as the embryo and the substance are uh, in the lycopodium and collagen. The hypobasal half of the embryo gives rise to the foot and to root. So the root is quadrant in demarcated in the embryo, unlike the lycopodium, where the root arises exogenously. And the upper epibasal means hypobasal is then root and foot. Epibasal, the epibasal half form the apical cell of the stem and cell lateral to the produce the first leaf sheath. The stem of the embryo grows into the primary limited growth. Finally, one secondary branch grows down to the soil and develops into the perennial rhizome or unlimited growth. Again. So here the life cycle of the equisetum. Here, first of all, the equisetum profile to develop to the stabulus or sporangiophores. The sporangiophores to bear the sporangia. The sporangia inside the sporangia spore mother cells are there. Spore mother cells they are undergoes to the meiotic division to produce the spore. Four spores are there. The spores again germinated in the suitable or favorable condition. They are to develop as a prothallus or either gametophyte. So in that gametophyte, which bear the antheridia and archegonia, they are to produce the respective gametes. After gametes development to release and to fuse to form a zygote. So the zygote to develop as a some perichylial and epibasal hypobasal cell that to develop as a embryo. So after that embryo to develop as a sporophyte. Okay, clear? Okay, understood. Equisetum. Is
Okay,
audible my voice? Okay. So, so
sir.
you keep it. Okay. So that keep it my as a cost. Again, it is recorded now. Vijayalakshmi garu.
Yes, sir.
What? It is
sir.
recorded. A coast ఇవ్వండి అక్కడ నాకు. Please keep it coast in my name. Hmm? So the next topic is the next topic is the telome theory. Okay. So studies on the paleobotanist have thrown light not only the interrelationship of the various groups with the pteridophytes, but also on the way to which is the plants may have been derived from these means thoughts. But there are no fossil which near clearly link pteridophytes with their possible ancestors. These discussions still take place on the whether the pteridophyte evolved directly from algae or from bryophytes, whether in this case the monophyletic or polyphyletic theories are there to explain the evolution of these pteridophytes. But the Precambrian period, there seems to a little hope agreement of this matter. So, in the xylem and phloem pericycle and sometimes medullary rays and pith is also present. They are called as a steel. So, here in Van Thegem and Duleit is developed a theory called stellar theory. This is used as a steel, collectively since I mentioned in the steel, not only made up of the xylem and phloem, but the tissues like pericycle, vascular rays, and pith is also associated with to formed as a steel. So, according to the cortex and steel, the fundamental parts of the shoot, both these parts are separated from each other by the endodermis. This is actually the basis of the stellar theory. So, according to this theory, stellar theory, here, so there is no different growth anatomy of the primary bodies, stem and roots, because both the steel surrounded by the cortex is present. So it is believed the large leaves of fern originated phylogenetically due to the specialization development for the dichotomous branching system. This concept stem like the nature of the megaphilus was first suggested by the Linger. So Lingener, which is on Bower, after they are developed as the telome theory, and Zimmerman is also will explain about these telome theory. So the telome theory was proposed by the Zimmerman in 1930, later elaborated by him. According to these vascular plant evolved from the very simple and leafless and dichotomously branched ancestor like Rhinia. Such plants is made up of steels and fertile branches are there. So he named the simple term of the dichotomous branching as a telome. Later on, the Foster and Gifford is defined as the telome, unlimited terminal portion of the dichotomizing axis. So this one called as a telome. So ultimate terminal portion of the dichotomizing axis. So it may be is not bear the sporangia, may be bear or may not bear. It bears sporangia at the tip. It is called as a fertile telome. Not bear, that is the sterile telome. So, according to Zimmerman, the fertile telome had sporangia at their tip. Those are had no sporangia were named as the sterile telome or phylloid. So, as the telomes are ultimate part of the branches, and the portion of branching before its bifurcation is named as the mesome. So during the source of evaluation of the dichotomous branching changes into sympodial branching due to the fusion of two or more telomes. Such fused portion of two or more telomes called as the syntelomes. According to telome theory of Zimmermann, the primitive pteridophyte evolved from unicellular member of green algae. These green algae members is divided into directions, became the parenchymatous and developed the meristematic tissue which give rise to erect branches. Later on, the sporophytic and gametophytic generations develop into the plant body. So, the conducting tissue developed into the dichotomous branching of sporophyte. The primitive vascular cryptogams were evolved. Um, the latter, they are the sporophytic and gametophytic generation develops in the plant body. The conducting tissue developed in the dichotomous branch of the sporophyte. The primary primitive vascular cryptogam were evolved such as algal ancestors during the upper Silurian and lower Middle Devonian period. Mhm. So, the present body of the primitive vascular cryptogams was simple dichotomously branched without any root or leaf and had a simple protostele. Instead of the roots of rhizomes were present, some of the branches of such as sporophytic plant developed sporangia at their tips and representing the fertile telome. So here elementary processes. Here there are the lower vascular cryptogamic plants which are the evolved into higher cryptogams through the independent evolutionary line of the Lycopodia, Phenopsida and Pteropsida. Here, the elementary process involved in the overtopping and planation, genesis, reduction, and curvation. So these are there. Here, overtapping means this process occurs during the formation of leaves from the axis. So the out of the two equal dichotomous branches, one branch is large and mechanically stronger. This larger branch of the axis, while shorter one form the lateral axis or leaves. So this one is the overtapping, overtapping process and planation. Here it occurs in the system of dichotomous. This process results arrangement of the telome and mesomes in the same plants. Then syngenesis, this is a process called and webbing means joining or attachment. So in this process, the fusion of telomes and mesosomes takes place with the development of parenchymatous tissue. This implement process explains the origin and evolution of the leaf as well as steel of the stem. The syngenesis result in the formation of leaf with open dichotomous venation, pinnate venation, and reticulate venation, and complex steel tissue. In selaginella stem, the polystylic condition occur as the result of simple parenchymatous webbing. So another one is the reduction. Here, this reduction of the telome takes place in the zoophytic plant, where the leaves are microphyllous like the Lycophyta and Spinophyta. In such cases, the leaves become reduced like a scales or needle-like shape. So another one is the curvation. These fertile became curved, one bend to downwards. So this is incurvation and recurvation here. This process downwards shifting of the sporangia occurs from the terminal or ventral surface of the leaf, like part of the member of the Pteridophyta. That is incurvation or recurvation here. It was defined by the word law as a process where the downward bending of the sporangia occur along the sporangial stalk as the Spinophyta. So this is the telome theory to explain the okay. The next one is the origin of the sporophylls. Okay.
Hello?
sir was leaving.
Why?
I don't know.
Hello? Hello, madam.
Hello?
Class is not there, na?
I mean, we don't know.
Sir, number is there. Please message him. Any
What happened? The class is not going on.